Welcome back for another deep dive. This time, you've really outdone yourselves with the source material.
It is quite a collection.
Nobel lectures, research articles, I even saw a historical account in there.
Yeah.
It's really like we've got a backstage pass to some of the biggest moments in science. So,
I think it's going to be a good one.
yeah, where do we even begin with all this? Well, I guess our mission, as always, is to try to pull out those key insights, those aha moments that really changed our understanding. And also how these discoveries connect, right?
Yeah.
Across different fields, because we've got chemistry, biology, physics represented.
And across time.
And across time.
Yeah.
Decades of research here.
Yeah.
So much to unpack, ready to dive in.
Absolutely. Let's go.
All right. Let's start with E.J. Corey. This is from his Nobel lecture, NBL 297.pdf. He's talking about his work on prostaglandins.
Mm-hmm.
Now, prostaglandins, you might not be familiar with them, but they're these really important molecules involved in all sorts of things in your body, like inflammation and blood pressure regulation.
They're actually lipids, which are fats.
Lipids. Okay.
And they act like hormones.
Okay.
But unlike hormones, they work locally in the tissues where they're produced.
I see.
They have a wide range of effects, which makes them really fascinating to study.
And Corey, what did he do?
Well, he figured out how to synthesize them in the lab.
Okay.
Which was a huge deal because these are really complex molecules.
Ah.
It's not easy to just stitch those atoms together in the right way.
So he basically created a recipe for making prostaglandins.
Yeah, that's a good way to put it. And his work wasn't just about making these specific molecules. It was about developing new strategies for building any molecule, no matter how complex.
And he actually predicted that chemical synthesis in the future will be even more advanced than what he was able to achieve.
Right. He saw this as just the beginning. He had this vision of being able to create any molecule imaginable, which could revolutionize medicine, material science, all sorts of fields.
So this isn't just looking back, it's like we're getting a glimpse into the future.
Absolutely.
Of what might be possible.
It's pretty inspiring when you think about it.
Yeah, really is. But before we get too far ahead of ourselves, I think it's important to understand the foundation that Corey's work was built on, right?
Of course.
Because it didn't just come out of nowhere.
No, it's all connected. And actually, one of the key figures in this story is Hermann Staudinger,
Okay.
who won the Nobel Prize for his work on macromolecules. And we have an excerpt from his lecture here, NBL300.pdf.
All right, so this is early 20th century. What was going on in science at that point? What did they even know about macromolecules?
Well, that's the thing. They didn't really know much at all.
Okay.
I mean, they knew that substances like cellulose and rubber were different
Right.
from smaller molecules like water or alcohol.
Uh-huh.
But they didn't really understand why.
Okay.
The prevailing theory was that these substances were just loose collections of small molecules held together by some kind of mysterious forces. They called them colloids.
Colloids.
Yeah. And there was a lot of debate about whether these were actually true molecules or just aggregates of smaller units.
So kind of like a puzzle that they didn't have all the pieces for.
Exactly. And that's where Staudinger comes in. He challenged this whole idea.
Okay.
He argued that these substances were actually macromolecules,
Yeah.
single, giant molecules with long chain-like structures.
Wow. So it's like the difference between a pile of bricks and a skyscraper, right?
Yeah, that's a great analogy.
Both made of bricks, but the way they're structured makes all the difference.
Exactly. And Staudinger's work was met with a lot of skepticism at first.
Really?
Oh, yeah, a lot of resistance from the scientific community.
Huh.
But he was persistent.
Okay.
He kept doing experiments, gathering evidence, and eventually his ideas won out.
Well, good for him.
Hmm.
So how did this change our understanding of materials?
Oh, it was a complete revolution. Think about it. Plastics, synthetic fabrics, DNA, even the proteins that Corey was working with later on, they're all macromolecules.
Oh, I see.
Staudinger's work laid the foundation for understanding these incredibly important substances.
It's amazing how one scientist's idea can have such a ripple effect,
It really is.
shaping the world we live in.
And it just goes to show how scientific progress builds upon itself.
Yeah.
Each discovery opening up new possibilities for the next.
It's all connected.
Exactly.
All right. Well, from the world of macromolecules and chemical synthesis, let's shift gears a bit. Let's talk about bioluminescence. This is the ability of living organisms to produce light, like fireflies or certain jellyfish.
Right.
And that brings us to Osamu Shimomura, who won the Nobel Prize for his work on the jellyfish Echoria victoria. This is from his Nobel lecture, NBL302.pdf. Now, I have to admit, when I think of jellyfish, I think of those mesmerizing creatures pulsing with light in the ocean.
Beautiful, right,
Beautiful. I don't immediately think scientific breakthrough.
right.
But what was it about Shimomura's work that was so groundbreaking?
Well, he dedicated years to isolating and characterizing the proteins responsible for bioluminescence in these jellyfish,
Okay.
specifically acherin and green fluorescent protein, or GFP as it's known.
GFP.
And this was incredibly painstaking work. Imagine trying to extract these delicate proteins from tons and tons of jellyfish. It's a monumental effort.
It sounds like it.
But he persisted,
Okay.
and his meticulous research led to a really surprising discovery. He found that the luminescence systems of two completely different jellyfish species, Acheria and Cypridina, were actually linked.
Okay, how so?
Well, they both relied on a molecule called coelenterazine, even though their light-emitting proteins were different.
So it's like finding out that two cars that look completely different on the outside actually share the same engine under the hood.
That's a perfect analogy. And this wasn't just a curiosity. It revealed a deeper evolutionary connection between these seemingly unrelated species and opened up new avenues for understanding how bioluminescence works.
It makes you wonder what other hidden connections are out there in the natural world, just waiting to be discovered. Now, speaking of intricate biological systems, this leads us to Christian Anfinsen and his work on protein folding. This is NBL303.pdf. Remember those complex light-emitting proteins that Shimomura isolated?
Aye.
Well, Anfinsen was trying to figure out how proteins in general fold into these precise three-dimensional shapes that determine their function.
It's like a molecular origami project.
It is. You've got these long chains of amino acids, and they somehow know how to fold themselves into these incredibly intricate structures.
It's one of the most fundamental questions in biology. How does a protein sequence determine its structure?
Right. Is there like some tiny instruction manual hidden inside each protein? How do they do it?
Well, Anfinsen's work showed that the instructions are actually encoded in the amino acid sequence itself.
Really?
He demonstrated this through a process called reversible denaturation. He found that some proteins can be unfolded, basically scrambled up, and then, remarkably, refold themselves back into their functional shapes.
So it's like crumpling up a piece of origami and then watching it magically unfold itself back into a perfect crane. That's incredible. So the information for how a protein folds is built into the sequence of its amino acids.
Right.
It's like a self-assembling puzzle.
Exactly. And this discovery had profound implications for understanding how proteins work, how they evolve, and even how we might design new proteins with specific tasks.
It's pretty mind-blowing. All right, now buckle up because we're about to shift gears again.
Okay.
From the intricate world of protein folding to the realm of analytical chemistry, where scientists develop techniques to separate, identify, and quantify the components of complex mixtures.
It's like detective work at the molecular level.
It is, and this brings us to RLM Singe and his work on partition chromatography. This is described in NBL304.pdf. Now, chromatography, this technique, revolutionized the way scientists analyze mixtures, particularly proteins. Imagine trying to understand a symphony by listening to all the instruments playing at once.
Chaos.
It would be impossible, right?
Right, you couldn't make sense of it.
Chromatography is like separating out those instruments so you can hear each one distinctly. So how does it work? What's the magic behind this separation?
It's based on the principle that different molecules have different affinities for two phases, a stationary phase and a mobile phase. Imagine a mixture flowing through a column packed with a material. Some molecules will stick to the material more strongly, while others will be carried along more quickly by the flow.
I see.
And this difference in movement allows them to be separated.
It's like a race where each runner has a different strategy for navigating the course. Some cling to the edges while others sprint ahead.
Exactly.
So who wins?
Well, it depends on what you're looking for. Sometimes you want the fastest molecule, sometimes the slowest. The point is, you can separate them based on their properties.
And this technique has had a huge impact on so many fields.
Oh yeah, from medicine to environmental science to forensics, it's used everywhere.
Now, Singe actually collaborated with Hedley Marston, an expert in animal nutrition. This is a great example of how fundamental research can have unexpected practical applications.
Right. Marston saw the potential of chromatography for analyzing the protein composition of wool.
Okay.
By understanding which amino acids were present and in what proportions, they could gain insights into the nutritional needs of sheep.
Wow, so it's not just about separating molecules. It's about understanding what those molecules tell us about the biological systems they come from. And what did they discover about wool?
Well, one of the key findings, not just from their work, but from the wider use of chromatography, was that most proteins are actually composed of just the 20 known amino acids.
Really? It's incredible to think that such complexity arises from a limited set of building blocks.
That's like building an entire language with just 26 letters.
Exactly. Now, speaking of using sophisticated analytical techniques to unravel biological mysteries, let's talk about Soon Bergstrom and his research on prostaglandins. This is described in NBL 305.pdf. Remember we talked about Corey synthesizing prostaglandins in the lab?
Right.
Well, Bergstrom was trying to figure out how these important molecules are made in the body.
It's like reverse engineering nature's recipe.
Exactly. But it wasn't easy, right?
Not at all. These molecules are often very reactive and short-lived, like fleeting shadows, difficult to capture and study.
I see.
Bergstrom had to develop some really innovative experimental approaches to track these elusive intermediates in the prostaglandin synthesis pathway.
So it's like trying to catch a glimpse of a rare bird in flight. You need the right tools and a lot of patience.
Exactly. And through these meticulous efforts, he discovered some really interesting things.
Like what?
Well, he identified some unstable intermediates, including a mysterious molecule dubbed RCS that played a crucial role in the transformation of certain fatty acids into prostaglandins.
RCS, huh? It's amazing how scientists can piece together these complex biological puzzles step by step,
Yeah.
uncovering the hidden mechanisms that drive life. But before we go any further down that path, I think it's time for another shift in perspective.
All right. Where are we headed now?
We're going subatomic.
Okay.
Let's talk about isotopes. These are variations of elements with different numbers of neutrons.
Right.
And this brings us to Harold Urey's discovery of deuterium, the heavy isotope of hydrogen. This is NBL 314.pdf. Now, I have to admit, isotopes always sounded kind of intimidating to me.
They get a bad rap, I think.
Yeah, a little bit. What's so special about deuterium? Why is it such a big deal?
Well, before Urey's work, everyone thought hydrogen only existed in one form,
Mm-hmm.
with one proton and one electron.
Right.
But Urey and his team, they meticulously concentrated liquid hydrogen
Right.
and then analyzed it using spectroscopy.
Okay.
This is a technique that allows you to identify elements based on the light they emit or absorb.
So it's like using a prism to separate light into its different colors.
Exactly. But instead of colors, they were looking for the unique signature of deuterium.
And did they find it?
They did. To everyone's surprise, they found a faint spectral line that didn't correspond to ordinary hydrogen. That line was the fingerprint of deuterium, a heavier version of hydrogen with an extra neutron in its nucleus.
That's incredible. It's like finding a secret message hidden within the light itself. So this wasn't just a scientific curiosity.
No, not at all. This discovery revolutionized our understanding of nuclear physics, chemistry, even cosmology. Deuterium plays a crucial role in nuclear reactions. It can be used to track chemical reactions and provides insights into the processes that occurred in stars.
Wow, it's amazing how something as seemingly simple as an extra neutron can have such profound implications. It's a reminder that even the smallest details can hold incredible secrets.
Absolutely. And speaking of uncovering secrets, are you ready to dive into the world of semiconductors and the invention that revolutionized electronics?
You bet. Lead the way.
All right then, let's explore John Bardine's Nobel lecture on the development of the transistor. This NBL 308.pdf. Now, this tiny device transformed the way we live, work, and communicate, paving the way for computers, smartphones, all sorts of electronic gadgets we take for granted today.
Yeah.
But before the transistor, electronics were bulky, inefficient, and unreliable.
It's hard to imagine a world without transistors.
Right.
They're literally everywhere, but I have to admit, I have no idea how they actually work.
Well, to understand the transistor, we need to delve into the worlds of semiconductors.
Okay.
These are materials like silicon and germanium that have electrical conductivity somewhere between that of a conductor, like copper, and an insulator, like rubber.
So it's like a dimmer switch for electricity. You can control how much current flows through.
That's a good way to think about it. And what makes semiconductors so special is that their conductivity can be controlled.
Okay, how?
Well, by adding impurities, called dopants,
Dopants. Oh,
or
okay.
by applying an electric field. So you can fine-tune their behavior.
So we're doping semiconductors with impurities. This is where it gets a bit mind-boggling for me.
Well, it's all about understanding how electrons behave in these materials.
Okay.
The key lies in concepts like holes, which are essentially the absence of electrons.
Holes?
Yeah, like a missing tooth in a row. and donors and acceptors, which are impurities that can donate or accept electrons, respectively.
So it's like a game of musical chairs, with electrons hopping between different energy levels within the semiconductor.
Yeah, that's a good visualization.
Yeah.
And by manipulating these electron movements, Bardeen and his colleagues were able to create the transistor, a device that could amplify and switch electronic signals.
So it's not just about controlling the flow of electrons, it's about using that control to process information, which is the foundation of all modern electronics.
Exactly. And this breakthrough paved the way for the information age.
It's amazing how understanding the behavior of electrons at a fundamental level can lead to such transformative technological innovations.
It really is. It's a testament to the power of scientific curiosity and the interconnectedness of different fields of knowledge.
And speaking of pushing the boundaries of knowledge, let's take another leap. This time from the world of solid-state physics to the realm of subatomic particles.
Okay.
This brings us to Masatoshi Koshiba and his work on neutrino oscillations. This is described in NBL 309.pdf. Now, neutrinos are incredibly tiny, elusive particles that rarely interact with matter.
They're like ghosts passing through walls.
Exactly. They're incredibly hard to detect.
And what did Koshiba discover about them?
Well, he used this massive detector called Super Kamiokande in Japan to study neutrinos coming from the sun.
Okay.
And his research provided really compelling evidence that these ghostly particles actually change their identities as they travel through space.
Wait, what? So these neutrinos are shape-shifting? In a way, yes. It's a phenomenon called neutrino oscillations.
Neutrino oscillations.
Imagine you have a bowl of 3 different flavors of ice cream: chocolate, vanilla, and strawberry.
Okay, I'm following.
As you're eating, you notice that the flavors seem to change, even though you didn't mix them.
So it's like a cosmic magic trick. But seriously, what does this tell us about the universe? Why is this discovery so important?
Well, it challenged the standard model of particle physics, which initially assumed that neutrinos had no mass and couldn't change flavors.
I see.
Koshiba's work forced physicists to revise their understanding of these fundamental particles. It opened up all sorts of new questions about the nature of matter and the evolution of the universe.
So even our most fundamental theories are always subject to revision. It's a never-ending quest for knowledge. But wait, just when you thought we couldn't get any smaller, we're zooming in on single electrons.
This is getting really wild.
Hans Demelt's work, NBL 306.pdf, is all about isolating and measuring the properties of these tiny particles. And he did this with incredible precision.
Yeah, his experiments are legendary.
This is where the line between science and magic really starts to blur.
I agree. He used a technique called geonium, using electromagnetic fields to trap a single electron in a vacuum.
So they're holding a single electron in a force field and studying it like a tiny captive butterfly. But why go through all that trouble just to study one electron?
Well, by isolating a single electron, you can eliminate the noise and interference from other particles, which allows you to make extremely precise measurements of its properties.
I see.
And those measurements were crucial for testing fundamental physics theories.
So even something as small as an electron can hold profound clues about the nature of reality.
It really can.
It's incredible.
It's a testament to human ingenuity and the relentless pursuit of knowledge.
Yeah.
But our exploration doesn't end here. There's still much more to uncover.
There always is.
From the intricacies of chemical bonds to the fascinating world of macromolecular transformations, the journey continues.
Sounds like we've got our work cut out for us.
We do.
Are you ready to dive deeper?
Absolutely. Bring it on.
All right, let's do it.
Okay, so we've seen how scientists can build complex molecules, how proteins fold into those intricate shapes, and how we can separate and analyze mixtures with incredible precision.
It's amazing how far science has come, but I feel like we're just scratching the surface.
Oh, we are. There's so much more to explore. And our next stop takes us to the heart of chemistry, to those fundamental interactions between molecules.
Okay.
Specifically, we're going to look at electron transfer reactions.
Electron transfer reactions.
Yeah, these are reactions where electrons jump between molecules,
Okay.
and they're involved in countless processes, from photosynthesis to the way batteries work.
Wow, sounds important.
It is, and Henry Taube won the Nobel Prize for his work in this field.
All right. What did he discover?
Well, his research, which is described in NBL 317.pdf, delved into a rather heated debate at the time about the speed of these electron transfers.
Ah, a debate.
Yeah, there was a lot of back and forth, particularly about self-exchange reactions.
Self-exchange reactions, what are those?
So these are reactions where an electron is transferred between 2 identical molecules.
Yeah.
one with a positive charge and one neutral.
Okay.
And it's tricky to measure because it's like trying to figure out how quickly a dancer switches places with their reflection in a mirror.
Ah, I see what you mean. So scientists were arguing about how fast these electron swaps were happening.
Exactly. There was this controversy surrounding the rate of the iron 3 plus 2 plus self-exchange reaction. Some thought it was incredibly slow, others argued it was much faster.
And what did Taub find?
Well, his work, along with some definitive measurements by R.A. Dodson, helped settle the debate and provided key insights into how electron transfer actually works.
So how does it work? How do these electrons actually make the jump?
Well, it turns out there are two main mechanisms: outer sphere and inner sphere electron transfer.
Two mechanisms.
Yeah, outer sphere is kind of like a long-distance relationship. The molecules stay separate, and the electron sort of tunnels between them. Inner sphere, on the other hand, is more like building a bridge. The molecules form a direct connection, and the electron walks across.
Interesting. So is one mechanism better than the other?
Not necessarily. It really depends on the specific molecules involved in their environment. Taub's work helped us understand these different pathways and the factors that influence which one is favored.
So it's not just about knowing that electrons can hop between molecules. It's about understanding the choreography of that jump, the steps involved, and the factors that influence the dance.
Precisely. And this understanding has implications for many areas of science, from developing more efficient solar cells to understanding how enzymes catalyze reactions in your body.
Speaking of understanding how molecules interact, this brings us to Kenichi Fukui and his work on frontier orbital theory. This is NBL 320.pdf. Now, I have to admit, orbitals in quantum chemistry always felt a bit intimidating to me.
It can be a bit daunting, but the core idea is actually quite elegant. Fukui's theory focuses on these special orbitals called HOMO and LUMO.
HOMO and LUMO, those ring a bell. Remind me what those stand for again.
Sure. HOMO is the highest occupied molecular orbital, and LUMO is the lowest unoccupied molecular orbital.
Okay, so we've got these special orbitals, but what makes them so important? Why are they the stars of the show?
Well, think of them as the matchmakers of the molecular world.
Matchmakers.
The HOMO of one molecule and the LUMO of another are the orbitals most likely to interact.
Okay.
They determine how easily molecules can share or transfer electrons, which influences the types of bonds that can form and the pathways of chemical reactions.
So if these orbitals are compatible, the molecules can click together and react.
That's a good way to put it. Fukui's theory provides a really powerful framework for understanding and predicting chemical reactivity.
So it's like having a blueprint that helps us understand how molecules will behave.
Exactly. And Fukui didn't stop there. He actually extended his work to polymers, catalysts, and other complex systems.
So he took this theory and applied it to the real world.
Right. And his insights have had a huge impact on fields like organic synthesis and materials science, helping researchers design new molecules and materials with specific properties. It's like being able to tailor-make molecules for specific tasks.
It's amazing how these fundamental theories can bridge the gap between the microscopic world of atoms and molecules and the macroscopic world of materials and technologies that we use every day. But I feel like we're due for another shift in gears, don't you?
Absolutely. Let's keep moving. Where to next?
How about from orbitals and reactivity to the realm of macromolecules?
Sounds good to me.
This brings us to Yves Chauvin and his research on olefin metathesis. This is described in NBL 323.pdf. Now, olefin metathesis, this reaction involves rearranging the double bonds in molecules.
Right. It's a bit like shuffling a deck of cards, but with atoms.
Okay, shuffling atoms around, that sounds fun. But seriously, what's so special about this reaction? Why is this molecular shuffling so important?
Well, it turns out that olefin metathesis is incredibly useful for building new molecules, especially polymers. It's like having a molecular construction kit that allows you to snap together different building blocks in a very controlled way.
So it's not just about breaking and forming bonds, it's about strategically rearranging them to create new structures.
Exactly. And Chauvin's work was groundbreaking because he proposed a mechanism for this reaction that involved these things called metal-carbene intermediates.
Metal carbenes, that sounds a little intense.
They are pretty reactive. They act like tiny molecular scissors, snipping and rejoining double bonds. But when Chauvin first proposed this mechanism, it wasn't readily accepted. There was this kind of divide at the time between homogeneous catalysis, which typically occurs in a solution, and heterogeneous catalysis, which often happens on a solid surface.
Okay, so why the hesitation? Were scientists just resistant to new ideas?
Well, It was more that Chauvin's mechanism seemed to blur the lines between these two types of catalysis, and people were a bit stuck in their ways of thinking.
I see. So it was a challenge to the existing paradigms.
Exactly. His work showed how similar principles could operate in both homogeneous and heterogeneous systems. It bridged the gap.
So it's like discovering that two different styles of dance actually share some fundamental steps.
Yeah, that's a great way to put it. And this realization had big implications. It opened up new avenues for developing more efficient and versatile catalysts.
And did this lead to any new breakthroughs?
Oh, absolutely. Chauvin's insights paved the way for the development of incredibly powerful ruthenium-based metathesis catalysts by Robert Grubbs.
Okay, so Grubbs took Chauvin's theoretical framework and turned it into a practical tool.
Exactly. And these ruthenium catalysts are remarkable. They're very versatile, tolerating a wide range of functional groups and reaction conditions. They've revolutionized fields like polymer chemistry and organic synthesis, making it possible to create new materials and molecules that were previously very difficult, if not impossible, to make.
It's like having a set of molecular tools that allows you to build almost anything you can imagine. And how about applications? Did these catalysts lead to any real-world advancements?
Oh, tons. They're used in everything from pharmaceuticals to advanced materials to green chemistry. It's a real success story for how fundamental research can lead to practical applications that benefit society.
It's amazing to see how these discoveries are shaping the world around us. All right, well, from molecular construction kits and powerful catalysts, let's turn our attention to something even more fundamental, the incredible world of DNA and protein synthesis.
The essence of life.
Exactly. This brings us to Marshall Nierenberg and his groundbreaking work on deciphering the genetic code. This is described in NBL324.pdf. Now, Nierenberg, he was essentially cracking the code of life, figuring out how the sequence of bases in DNA translates into the sequence of amino acids that make up proteins.
It's like deciphering a secret language hidden within our cells.
It is. But how did he even begin to tackle such a complex puzzle? I mean, where do you even start?
Well, Nierenberg and his team developed a brilliant approach. They created a cell-free system that could synthesize proteins.
Okay.
This allowed them to isolate the ribosomes, which are the cellular machinery responsible for protein synthesis, and feed them synthetic RNA templates with specific sequences.
So they were giving the ribosomes instructions written in the language of RNA and watching to see what proteins they produce.
Exactly. And by systematically testing different RNA sequences, they were able to figure out which amino acid each sequence coded for.
It's like they were cracking a code one letter at a time, painstakingly building a dictionary of the genetic language.
And eventually, they were able to decipher the entire genetic code, revealing the rules that govern how information flows from DNA to RNA to proteins.
It's a landmark achievement in biology. It's like finding the blueprint of life. But once we know the code, the next question is, how does it get copied? How does DNA replicate itself? And that brings us to Arthur Kornberg and his research, described in NBL346.pdf.
Right, so Kornberg's discovery of DNA polymerase was another huge step forward. This enzyme is the molecular machine responsible for faithfully copying DNA, ensuring that the genetic information is passed on accurately from one generation to the next.
So DNA polymerase is like the ultimate scribe, meticulously copying the book of life.
That's a great analogy. But it wasn't an easy task to figure out how this complex machinery worked. Kornberg and his team had to overcome some major challenges.
What were some of the hurdles they faced?
Well, imagine trying to study a tiny, intricate machine that's constantly moving and interacting with other molecules.
That doesn't sound easy.
It's like trying to repair a watch while it's still ticking. But they figured it out.
And what did they discover?
They were able to piece together the mechanism of DNA replication, revealing how DNA polymerase uses the existing DNA strands as templates to create new complementary strands. It's a beautiful example of the precision and efficiency of biological systems.
It really is. These discoveries, deciphering the genetic code and understanding DNA replication, have laid the foundation for modern molecular biology, genetics, biotechnology. They've revolutionized our understanding of life itself. It's incredible.
It is. And it just goes to show how seemingly disparate fields, from the intricate world of proteins and catalysts to the fundamental language of DNA, are all interconnected.
It's all part of the same story.
Exactly.
The story of life. But our journey through the world of scientific discovery isn't over yet.
Oh no, we're just getting started.
We still have more to explore, including the fascinating world of viruses and the intricate dance between pathogens and our immune system.
A microscopic battleground.
It is. Are you ready to delve deeper into this world?
Absolutely. Let's continue our deep dive.
All right, so we've gone from synthesizing complex molecules to deciphering the genetic code, from harnessing the power of electrons to understanding the foundations of immunity. It's amazing how much ground we've covered, but I feel like there's always more to uncover.
Absolutely, there's always more to explore. And our next stop takes us to the microscopic battleground of viruses and immunity.
Okay, viruses. Those tiny invaders that hijack our cells and cause all sorts of havoc, I have to admit they're fascinating and a bit terrifying.
They are indeed remarkable entities. And to understand how they work, we need to go back to John Ender's groundbreaking work on cultivating poliovirus in human tissue cultures, described in NBL 326.pdf.
Okay.
This was a major breakthrough that paved the way for the development of the polio vaccine.
So before Ender's work, what were the challenges of studying viruses?
Well, viruses are these obligate intracellular parasites, meaning they can only replicate inside living cells.
So they need a host to survive.
Exactly, which made it incredibly difficult to study them in isolation.
So it's like trying to study a fish that can only survive in water. You need a special environment to keep it alive and observe its behavior.
That's a great analogy. Enders and his colleagues figured out how to grow poliovirus in human tissue cultures, essentially creating a miniature environment where the virus could thrive and be studied.
It's like building a tiny aquarium for a virus.
Exactly.
But how did this lead to the development of a vaccine?
Well, by being able to grow the virus in the lab, researchers could study its properties in detail, understand how it infected cells, and most importantly, develop ways to weaken or inactivate it.
So they could essentially disarm it.
Right, which was crucial for creating a vaccine that could trigger an immune response without causing the disease.
It's incredible how something as seemingly simple as finding a way to grow a virus outside of the body can have such a profound impact on human health.
It's a testament to the ingenuity and persistence of researchers. And speaking of our body's defenses, this brings us to Jean Dausset's research on the major histocompatibility complex, or MHC, described in NBL 337.pdf. This is where the story of viruses intersects with the fascinating world of immunology.
Okay, the MHC, I remember hearing about this in biology class, but it always seems so complex. Can you remind me what it is and why it's so important?
Sure. The MHC is a set of genes that code for proteins on the surface of our cells. These proteins act like ID cards.
ID cards.
Yeah, allowing our immune system to distinguish between our own cells and foreign invaders like viruses or bacteria.
So it's like a cellular security system that checks for the right credentials.
That's a great way to think about it. And what Dausset discovered was that the MHC is incredibly diverse. Each individual has a unique set of MHC genes, which means each person's immune system has a slightly different way of recognizing threats.
Oh, wow. So it's like everyone has a unique fingerprint, but for their immune system.
Exactly. And this diversity is crucial for our ability to fight off a wide range of pathogens. But it also poses a challenge for things like organ transplantation.
Ah, I see. Because if the donor's MHC doesn't match the recipient's MHC, the immune system might see the transplanted organ as a foreign invader and attack it.
Precisely. That's why MHC matching is so important for successful transplantation. Dausset's work revolutionized our understanding of transplantation, immune responses, and even autoimmune diseases where the immune system mistakenly attacks the body's own cells.
It's fascinating how these seemingly disparate fields, virology, immunology, transplantation, are all interconnected.
Absolutely. And Dausset's work raised a profound question. How does the MHC, with its incredible diversity, contribute to self-recognition, the ability of our immune system to distinguish between self and non-self?
It's like the MHC is a key that unlocks the mysteries of our immune system, revealing how it navigates this complex world of pathogens and maintains this delicate balance of protecting us without attacking our own bodies.
Indeed. Well, we've covered a lot of ground today, from synthesizing life-saving molecules to deciphering the language of DNA, from harnessing the power of electrons to understanding the complexities of immunity. We've seen how science has shaped our world in really profound ways.
And it's not just about the individual discoveries, it's about how they connect, how they build upon each other to create this ever-expanding tapestry of knowledge.
Exactly. Advancements in one field often spark breakthroughs in others. It's a constant reminder that science is not a collection of isolated facts, but rather a dynamic, interconnected web of ideas.
So as we wrap up this deep dive, I encourage you to think about the connections you've made, the questions that have been sparked, and the new paths of exploration that have opened up for you. Where will your deep dive take you? The pursuit of knowledge is a never-ending adventure.
It is, and the world is full of mysteries waiting to be unraveled.
Until next time, keep those minds curious.